3, 7, 13, 31, 37, and 73. The biblical and DNA signature. You will hear, in this video clip, Richard Dawkins make the statement that the creator of this world could ingeniously encode a signature in its design.
Suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer.
This numeric signature is encoded in the DNA molecule, Genesis, and the Gospel of John. The signatures are there for all to see, and this is what these presentations will explain. The signatures are 3, 7, 13, 31, 37, 73. The SETI project stands for the Search for Extraterrestrial Intelligence. SETI monitors radio signals supposedly sent by aliens, which the public believe must exist. No detection of aliens has been found. Some scientists say radio signals are an unlikely method to succeed because we ourselves have only transmitted radio waves for just over 100 years. Therefore, scientists believe there is a much better way of detection of unlocking the truth for extraterrestrial intelligence by examining the signature of the numerical molecular structure of DNA. The analysis of the numerical and algebraic structure of DNA reveals the invention of the decimal system and zero with positional arithmetic, that is, 100s, 10s, units of modern arithmetic, the sign of intelligence. The molecular structure in DNA shows the signature of the prime number 37 encoded in the molecule, and this discovery of 37, the scientists say, reveals a new reality. What reality? The scientists say it is the irrefutable evidence of extraterrestrial intelligence, as no natural random mechanism can produce this numerical creation. The scientists use words, whatever it was, to avoid God. To say God for this intelligent source is verboten. Why forbidden? If the evidence points in that direction, so be it. Please note, the scientists fail to mention the tens of thousands of molecular machines which are necessary to make the DNA operational as a chemical program that is self-reproductive, self-diagnostic, and self-repairing, but only under the control of the molecular machines of the cell. This alone proves intelligent design. These scientists say DNA is the most stable recording method known to man, and the structure is digital, meaning the instruction codes are discrete molecules. The scientists would prefer to use the evidence of DNA numerical analysis for the SETI project as a more fruitful method than radio detection. The mathematical DNA analysis is the summary of the work of Professor Vladimir Scherbak and Maxim Markov, as documented in their papers, peer-reviewed, which are Arithmetic Inside the Universal Genetic Code and the WOW Signal of the Terrestrial Genetic Code. Later, we will prove that these signatures in the arithmetic of DNA are the very same signatures in the arithmetic of the words and sentence structures in the key scriptures of Genesis 1:1, Deuteronomy 6:4, John 1:1. The scientists' research shows an intelligent structure, but the DNA does not tell us who that intelligence was. Scientists use the words whatever it was, but the numerics and algebraic structure in the scriptures clearly identifies who this intelligence was, and is the Lord Jesus Christ, the creator of the universe. In the beginning God, Hebrew, Elohim, created the heavens and the earth. We will explain simply the chemistry of the DNA and a video of the quantum atomic structure of the universe with a biblical numerical analysis and how they produce the signatures that are all encoded, that is, set in stone by the intelligent source. Now a strong reminder that this DNA evidence has not been produced by creationists, but the evolutionary mathematical biologists. This video gives the proof of the very same signatures encoded in Bible maths. The book 373, The Proof, Set in Stone, with booklets, DNA, the 70 Weeks of Daniel, and the 7 Unusual Numbers, gives detailed explanations of the arithmetic of the DNA signatures and Bible maths. You may be a little overwhelmed by the first half of this video, by the arithmetic, but don't give up, because later in the video, there are some interesting animations and interviews, including one with Richard Dawkins. A numerical analysis of the genetic coding revealed in the DNA of living cells and how it relates to the numerical structures in the biblical scriptures of Genesis chapter 1 verse 1, Deuteronomy chapter 6 verse 4, and the Gospel of John chapter 1 verse 1. Here is a very simplified image of a cell. Nucleolus. Nucleus. Chromatin. Nuclear envelope. Centriole. Vacuole. Microtubules. Lysosome. Golgi apparatus. Secretion released by exocytosis. Cytosol. Peroxisome. Smooth endoplasmis reticulum. Rough endoplasmis reticulum. Plasma membrane. Ribosomes. Microvilli. Mitochondrion. Microfilament. The image illustrates the incalculable machinery of the living cell, with its 100,000 proteins coded for by 20 amino acids. Metabolic pathways. This image shows the immense complexity of the chemical connections in a living cell. The next 2 slides will enlarge the diagram and show how infinitely complex the living cell is, and how the design defies anything that we could have imagined. The diagram shows the conversion of ADP to ATP by an ultra-fast rotating protein motor, which uses the energy from the food we eat, and this motor continuously makes ATP energy for all cell processes. Without this ADP to ATP conversion, then all life would cease to exist on our planet. The chemistry of the cell is infinitely more complex than can be shown in a diagram. The protein motor spins at 8,000 revolutions per minute, giving 24,000 ATP molecules per minute, which amounts to processing 50 to 75 kilograms per day. That is a person's own body weight. This part of the diagram shows the conversion in plants of ADP to ATP using the same protein motor, taking the sun's photons and via photosynthesis, providing the energy for the plant cells. Anybody can see that the cell is unbelievably more complex than anybody had conceived. We will now investigate how DNA works and its numerical signature. What is the genetic code, and how does it work? The genetic code carries all the instructions to build all life on this planet. These instructions are found in a gigantic molecule called DNA deoxyribonucleic acid. The information is passed from cell to cell in the reproductive process. The DNA molecule is composed of a basic unit called a nucleotide. The nucleotide is made of a sugar deoxyribose, item D, and a phosphate group, item P. All joined together on a nitrogenous base, item A. There are 4 nitrogenous bases in a DNA molecule: A, and G, and T, and C. The sugar D and the phosphate P bond together to form a backbone for the nucleotides to ride upon. The backbone carries no information at all. The DNA is in 2 separate halves that are bonded by the 4 nucleotides. The nucleotides only join together as shown, A with D and C with G. The unique pairing of only A with D and C with G enables the DNA to make a copy of itself when the 2 halves of the DNA are separated. It is the nucleotides that carry the instructions to build the living cell. The geometric structure of DNA is shaped as a double helix. This is a very elegant, ingenious, intelligent design. How is the information? The instructions conveyed on this double helix ladder. The bases are in groups of 3 letters called a codon. There are 64 possible permutations of 4 bases in groups of 3 letters. The instructions to make 20 amino acids are in the 3-letter coding. The 3 different groups of letters form individual words, and the groups of words form genetic sentences, proteins called genes. There are 3 billion base pairs in the human genome. Examples of codons are AAC, asparagine; TGT, cysteine; CTA, leucine; AAA, lysine; GTC, valine. Here is the list of the 20 amino acids: alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, triosine, valine. Now follows the 64 triplet codons and the amino acids they code for: isoleucine, methionine, threonine, aspartic acid, glutamic acid, glycine, valine, alanine, tyrosine, stop code, cysteine, stop tryptophan, phenylalanine, leucine, serine, histidine, glutamine, arginine, leucine, proline. The atomic structure of each amino acid is composed of a standard block, called B. A unique side chain, called the radical R, is then added to the standard block, which then forms each individual amino acid. Adding the radical makes valine. Adding the radical makes tyrosine. Adding the radical makes glutamic acid. This is the same standard block in all 20 amino acids. We will study the amino acids from the standpoint of arithmetic and prime numbers. We have changed the amino acids into numbers. This is achieved by counting the protons and neutrons in each amino acid. We split the count between the standard block B and the radical R. Alanine, cysteine, and serine. Remember the key numeric values in the scriptures. Genesis 1 verse 1 gives 7, 37, and 73. Deuteronomy 6 verse 4 gives 13, 26, 86, and 1118, with John verse 1 gives 3, 13, 31, 39, and 373. We will show that these very values have been encoded in the atoms of the amino acids of our DNA. All 19 of the amino acids in the standard block B have exactly 74 nucleons. Now 74 equals 2 by 37, and 37 is the signature of Genesis 1 verse 1, with value 2701. Which equals 37 by 73. The exception to this rule of 74 is proline, with 73 nucleons, but 73 is the other signature in Genesis 1 verse 1, and 74 is also the neighbor of 73. Inspecting the tables, you observe that the number of protons in the standard block B is 39, which equals 3 by 13. Now 39, and its reflection 93, is the signature of John 1:1, with value 3627, which is 39 by 93, and 13, the divine name of 26, which equals 2 by 13. Amazingly, we have 37 and 73, the very same signatures between scripture and DNA. To bring proline in line with the 19 amino acids, Scherbak uses a proton from the radical to give 39 protons, 74 nucleons, so Scherbak labels proline the activation key. First, the table shows the 20 amino acids and their molecular weights. The biblical value of Genesis 1 verse 1, in the beginning God created the heavens and the earth, is 37 multiplied by 73, which is 2701. Also Genesis 1 verse 2. And the Spirit of God was moving over the face of the waters, which has the value 37 by 37, which gives 1369. In integers, meaning whole numbers, the total molecular weight is 2738. This value is remarkably biblical, for Genesis 1 verse 1, 2701, which is 37 by 73, plus 37, which gives 2738, or twice Genesis 1 verse 2, 1369, which is 37 by 37, that is, 2738 is 2 by 37 by 37, which gives 2 by 1369. What follows is a numerical analysis of DNA by evolutionary biological scientists who are not creationists. Gratitude is given to Vladimir Scherbak for the following DNA studies. Scherbak's first symmetry. The table of codons for the amino acids can be split into two logical groups that code for the same amino acid, colored blue. The rest that do not code in groups of 4, colored green. The green and blue blocks are transformed by rotation of 180 degrees. The totals of the nucleons are again multiples of signature 37. 333 is 3 squared by 37, 592 is 4 squared by 37, 925 is 5 squared by 37. 3 squared plus 4 squared, 5 squared. Take note, Pythagoras. The Egyptian triangle 1110 is 30 by 37, 2220 is 60 by 37. Notice the equal split. The signature of Genesis is the same signature as DNA. Scherbak's second symmetry is formed by splitting the DNA codons into the purines and pyrimidines. The purines begin with codes A or G, the green block, and the pyrimidines begin with codes T or C, the blue block. Purines with A and G. Pyrimidines with T and C. The totals of the nucleons, multiples again, of signature 37. 629 is 17 by 37, 888 is 24 by 37, 1517 is 41 by 37. 814 is 22 by 37, 1628 is 44 by 37. The signature in Genesis 1:1 is 37. Now, 888, which is 24 by 37, is the value of the name Jesus in Greek. 814 is 2 by 407, which equals 2 by 11 by 37. The sixth word of Genesis 1:1 are split equally between the R and B blocks. Scherbak's third symmetry is formed in two ways, by columns, then by rows. First, add the outer columns. Outer columns 789 plus 888 equals 1677. Then second, the inner columns. Inner columns 654 plus 814 equals 1468. Total 1443 plus 1702 equals 3145. 1443 is 37 by 39, the double, yes, a double, signature. Signature 37 in Genesis 1:1 and signature 39 in John 1:1. 1702 is 46 by 37 from Genesis 1:1, the words, "In the beginning God and the earth." Now, 888 in the Greek language is the name of Jesus, and 888 is 24 by 37. 3145 is 85 by 37. Those who believe in evolution, random forces, would not have expected this non-random symmetry, and miraculously duplicated in Genesis 37 and John 39. Scherbak's third symmetry by rows. Add the nucleons for the green outer rows. Outer rows 654 plus 814 equals 1468. Then, add nucleons for the blue inner rows, and then appears a surprise. Inner rows 789 plus 888 equals 1677. Why a surprise? The same totals for the outer columns swap with the totals for the inner rows. Then the same totals for inner columns swap with the totals of the outer rows. Total 1443 plus 1702 equals 3145. 1443 is 39 by 37 in John 1:1 and Genesis 1:1. 1702 is 46 by 37 in Genesis 1:1. The signatures. In the beginning God and the earth. Now, 888 in the Greek language is the name of Jesus, and 888 is 24 by 37. 3145 is 85 by 37. The same phenomena of rows and columns interchanging with each other also occurs in the table of the atomic weights of the amino acids themselves. I find the following symmetry and analysis is astonishing, and should make any sane person sit up and pay serious attention to these indisputable facts. To all you hardened atheists, do not let this evidence fall on deaf ears. Just for a moment, let the thought of God's existence penetrate your consciousness, and your reward will be a new life, and you will know for certain that what you will believe in is based on fact. The table below shows the classification of amino acids by the evolutionary biologist, Professor Rakosevich. He is not a creationist. This classification was reviewed by his peers in scientific papers. Firstly, we inspect the column totals. 828.87 plus 503.5 gives 1332. Now 1332 is equal to 2 by 666, which is also equal to 18 by 37. Words 1 to 5 in Genesis 1, verse 1 is equal to 3 by 666. This is the numerical value of the words, "In the beginning God created the heavens." 755.78 plus 649.87 gives 1406, and 1406 equals 2 by 703, or 38 by 37. Now words 6 and 7 in Genesis 1, verse 1, where 703 is value of the words and the earth. Next, 666 plus 703 equals 1369 equals 37 times 37. The unique signature of 37 by 37, Genesis 1:2, and the Spirit of God was moving over the face of the waters. Secondly, split the center row total into two equal amounts, 297.85. Divide the center row value of 595.71 by 2. Now divide the table into two equal parts. We find, in whole numbers, the sum of each half of the table. 485.49 plus 585.70 plus 297.85 gives 1369.04, which is 1369 in whole numbers. Do the same for the bottom half of the table. The two halves have the same integer totals. 1369 plus 1369 equals 2738. The total of the table. The symmetrical split of the rows 2 by 1369 is mirrored by the split of the columns, which is twice 1369 equal to 666 plus 703, giving 2 by 1369. Genesis 1:1 equals 3 times 666 plus 703 equals 2701 equals 37 times 73, and reads, "In the beginning God created the heavens and the earth." Also Genesis 1:2 equals 1369 equals 37 times 37, which reads, "And the Spirit of God was moving over the face of the waters." The signature of 37 is the hallmark of the blueprint of life. Scherbak's fourth symmetry, those codons with only two identical bases, arranged between the purines and pyrimidines. 64 DNA codon triplets. The purines with two identical bases. Select the AA codons. Extracting the AA codons. Then, the AC codons. And finally, the AG codons. The AA group totals. We have the total of the radical in base A, giving 535 nucleons. Then the total of the standard block, giving 592 nucleons, with the total of base A of 1127 nucleons. Select the G codons. Extracting the T codons. Then, the C codons. And complete with the A codons. The G group totals. We have the total of the radical in base G, giving 464 nucleons, with the total radical of the purines of 999. Then the total of the standard block, giving 666 nucleons, with the total of the radical in base G of 1130 nucleons. The pyrimidines with two identical bases. Select the T codons. Extracting the C codons. Then, the A codons. Then finally, the G codons. The T group totals. We have the total of the radical in base D, giving 547 nucleons. Then the total of the standard block, giving 666 nucleons, with the total of the radical in base D of 1213 nucleons. Select the C codons. Extracting the T codons. Again, the A codons. Now completing the extraction, the G codons. The C group totals. We have the total of the radical in base C, giving 452 nucleons, and the total radical nucleons of 999. Then the total of the standard block, giving 666 nucleons, with the total of the radical in base C of 1118 nucleons. Examining the values generated by Scherbak's fourth symmetry. The totals for A and G. The totals for T and C. The final totals for A and G, T and C, 1998, 2590, and 4588. Signature 37, in DNA, God has authored in Genesis. 592 is 16 by 37. 1258 is 34 by 37. 2257 is 61 by 37. 1998 is 2 by 3 cubed by 37. 2590 is 7 by 10 by 37. 1332 is 2 squared by 3 squared by 37. 2331 is 3 squared by 7 by 37. 666 is 18 by 37, the integral part of the triangulation of Genesis 1 verse 1, and the Word was God, John 1 verse 1. 999 is 3 cubed by 37, the words, in the beginning God, and the words, created the heavens, Genesis 1 1. 1118 is 13 by 86, Deuteronomy 6 4, the Shema. The Lord our God is one Lord. 13 Hebrew equals 1. 86 Hebrew equals God. 1 13 God 86 amazing. Now the neighbor of 1118 is 1119, but 1119 is 3 by 373, and 373 equals Word. In Greek, John 114588 is 2 by 31 by 37. Elegantly, 31 and 37 are the signatures of John 1:1 and Genesis 1:1. The author of DNA is the author of Genesis and the Gospel of John. Scherbak's fifth symmetry: select those codons with 3 identical bases, split between the radical and the standard block, triple T. Triple A. Triple G. And triple
Now,
C. Select those codons with unique bases, that is, 3 different letters. The 3 different letters are rotated, then reflected. First set out the anti-clockwise codons. Rotate anti-clockwise. Start with the codon A, T, C. Next rotate anti-clockwise the letters, giving T, C, A. Finally rotate, giving C, A, T. Then the letters are mirror reflected. Reflect, then rotate clockwise. Now, with the reflected codon C, T, A. Next rotate clockwise the letters, giving A, C, T. Finally rotate, giving T, A, C. Rotate anti-clockwise. Start with the codon G, C, T. Next rotate anti-clockwise the letters, giving C, T, G. Finally rotate, giving T, G, C. Reflect, then rotate clockwise. Start with the codon T, C, G. Next rotate anti-clockwise the letters, giving G, T, C. Finally rotate, giving C, G, T. Rotate anti-clockwise. Start with the codon T, G, A. Next rotate anti-clockwise the letters, giving G, A, T. Finally rotate, giving A, T, G. Reflect, then rotate, clockwise. Start with the codon A, G, T. Next rotate anti-clockwise the letters, giving T, A, G. Finally rotate, giving G, T, A. Rotate anti-clockwise. Start with the codon C, A, G. Next rotate anti-clockwise the letters, giving A, G, C. Finally rotate, giving G, C, A. Reflect, then rotate, clockwise. Start with the codon G, A, C. Next rotate anti-clockwise the letters, giving C, G, A. Finally rotate, giving A, C, G. Now generate the nucleon totals. The radical of 703 nucleons and the standard block of 962 nucleons. Please note, the nucleon total values are the same for the clockwise and the anti-clockwise. The mathematical analysis is on the next page. The table has 703 nucleons in the radical, 962 nucleons in the standard block. Surprise! Signature 37 emerges yet again. Observe the total equal amounts of the nucleons in the radical and standard block between clock and anti-clockwise. Now 703 is 19 by 37, the value in Hebrew of the words and the earth, and the 37th triangular number, the center triangle, in Genesis 1 verse 1. Remember, DNA is created here on earth. (962) is 26 by 37. But 26 is the value of the divine name, the Tetragrammaton, Deuteronomy 6 verse 4. 1665 is 3 squared by 5 by 37. 3 squared and 3 cubed, 26, 31, 37, are all biblical signatures. The neighbor of 962 is 961, which is 31 by 31, John 1:1. The neighbor of 703 is 702, which equals 2 by 3 cubed by 26. The neighbor of 1665 is 1664, which equals 4 cubed times 26. We know that it is the Lord who created all life, that is the DNA on our planet, the Earth. At this point, I would like to call to your attention that these investigations were performed by Professor Scherbak and many others. These scientists are Scherbak, Department of Applied Mathematics, Al-Farabi Kazakh National University, Kazakhstan; Jean-Yves Boulay, France; Petrikov, I have
Russia.
examined the works of the above, and these mathematicians make no reference to the subject of biblical numerical analysis. The elegant numerical structure of DNA signature 37 contributes enormously to the debate about creation, signature 37 versus evolution, no signature.
Thanks are also given to Craig Pard-Cooper and Vernon Jenkins for their contribution to the subject. We will now investigate the works of Jean-Yves Boulay.
Inspecting the amino acids, it is clear any codon codes for the same amino acid ending in T and C. Or A and G. Except the codons ATA and ATG, and TGA and TGG as marked. These marked codons are exceptions to the above rule, so they are not included in the proton count. Boole calls these codons the rebel group. Boole brings the amino acid proline in line with the rest of the acids, making the proton count 39, giving a total proton count of 63. See the accompanying booklet extracting the proton totals as a 4x4 matrix. The total 4004 is 4 by 7 by 11 by 13. These are biblical prime signatures. Very promising. Total 4004 is 4 by 7 by 11 by 13, or 7 by 22 by 26, or 11 by 13 by 28. Then values 7, 28, 13, 26, give 7 words, 28 letters of Genesis 1 verse 1. 26, the divine name of Deuteronomy 6 verse 4. 13, the signature of John 1 verse 1
using the
and
matrix,
Deuteronomy
select
6 verse
a pair
4.
of one set of numbers, a pair of two sets of numbers, a pair of 4 sets of numbers, a pair of 8 sets of numbers, finally, all 16 numbers. 252 is 7 by 36, or 28 by 9. 224 is 7 by 32, or 28 by 8. 476 is 7 by 68, or 28 by 17. 448 is 7 by 64, or 28 by 16. 924 is 7 by 132, or 28 by 33. 1008 is 7 by 144, or 28 by 36. 1932 is 7 by 276, or 28 by 69. 2072 is 7 by 296, or 28 by 74. 4004 is 7 by 572, or 28 by 143. Encoded in Genesis 1 verse 1, the two signatures, 7 and 28, by design, 7 words and 28 letters. Genesis 1 verse 1 has 14 letters in the subject, and the verb, which is 2 by 7. The two objects have 14 letters, 7 each. The 3 nouns, 14, which is 2 by 7 letters, an amazing numerical structure in the protons. The proton matrix, viewed as shown. Now, 320 plus 192 plus 376 is 888, which is 24 by 37, the value of Jesus. Next, 888 plus 144 plus 192 plus 256 is 1480, which is 40 by 37, the value of Christ. The title of Jesus in the New Testament is the word value of 373. Therefore, 296 plus 128 plus 288 plus 300 plus 256 plus 224 is 1492, which is 4 by 373, the value of word. Now 1492 is the value of the prophecy of Isaiah, chapter 9, verse 6, Wonderful Counselor, Mighty God, Everlasting Father, Prince of Peace. Finally, 300 plus 160 plus 320 plus 252 is 1032, which is 12 by 86. And 86 is the Hebrew value of God. This symmetry gives an astonishing result about the deity. Also, the number of atoms in the 64 triplets of all the amino acids is 1147, which is 31 by 37. These values are direct factors of John 1 verse 1 and Genesis 1 verse 1. The symmetry, the yellow set, rotated anti-clockwise. And the pink set is shifted one column to the right. Adding the pink and yellow values gives 2236. (2236) is 13 by 172. Remember, 13, the signature of John 1:1. 2236 is 26 by 86. Now 26 is the value of the divine name. 26, the value of the Lord. 86, the value of God, Elohim. This next selection of the pink and yellow gives exactly the same value to 2236. This 26 and 86 spells out numerically the Lord God. 2236 is 2 by 1118. Now 1118 is 13 by 86. The Shema, one God. This next selection has a different but interesting value. The pink and yellow values this time give 2028. (2028) is 12 by 13 by 13. The signature 13, John 1:1. Plus the Shema in Deuteronomy 4:6. Once more, the next selection of the pink and yellow gives exactly the same value, 2028. 2028 is 3 by 26 by 26. 26, the divine name, the second time. 2028 is 2 by 26 by 39. 39 is the signature of John 1:1, 3 by 13. God's signature. The values of 13, 26, 39. For the protons. Genesis 1:1 to 7:01 is 37 by 73. Deuteronomy 6:4, 1118 is 13 by 86. John 1:1, 3627 is 39 by 93, and is also 3 by 13.
by 31 by 3.
All this symmetry in the amino acids, nucleon, and proton count proves beyond any reasonable doubt that random forces did not create life on Earth. Life on Earth was created by an intelligent source. To deny this shows almost invincible prejudice against anything spiritual.
What conclusions can we draw from the study of these numerical signatures in the DNA codons? It all shouts, non-random and created by God.
The most interesting phenomena are the numbers of creation, which are 7, 13, 26, 39, and especially 37, which are all demonstrated in the counting of the nucleons and protons in the 20 amino acids. You would not expect this kind of symmetry to exist in our universe, created by the blind forces of nature. The data shows even the invention of zero, and the existence of the decimal system. Remember, you have by design 10 fingers and 10 toes. The structure of the number of nucleons shows a mathematician at work.
Jesus said, "But the very hairs of your head are all numbered." The scriptures, Genesis 1 verse 1, John 1 verse 1, and Deuteronomy 6 verse 4, give the details of creation and the nature of the Godhead. They all agree numerically with the signatures displayed in the nucleons of the amino acids. I call to your attention again that these discoveries have been made by scientists who do not have the knowledge of the numerical signatures in the Bible. It is appreciated that it is difficult to follow all this arithmetic, but you can generally understand the point of these numerical signatures in both Scripture and the DNA codons. God is the author of both. This unknown information is the final nail in the coffin of the fairy tale for adults, evolution. This numerical data is scientific fact and obtained by the scientific method. The data can be reproduced from the DNA and the Bible, and tested by anyone.
The following video is published by Harvard University and shows the immense complexity of the inner workings of the living cell. You do not have to understand all that is shown, but you will be impressed by the enormous sophistication of the intelligent design. The obvious truth of intelligent design is to be found in the infinite detail of the living cell.
To repeat, I have added these videos so that you may see for yourself how infinitely complex is the design of the molecular machinery of the living cell. This has to be firmly grasped.
This video shows the immense complexity of the living cell, with the details of the leukocytes in the immune system.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Rafts' rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydrocarbon chains. Outside the rafts, kinks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparin sulfate proteoglycan on endothelial cells are presented to leukocyte 7 transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions. The inner leaflet of the bilayer has a very different composition than that of the outer leaflet. While some proteins traverse the membrane, others are either anchored into the inner leaflet by covalently attached fatty acid chains or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrin tetramers arranged into a hexagonal network are anchored by membrane proteins. This network forms the membrane skeleton that contributes to membrane stability and membrane protein distribution. The cytoskeleton is comprised of networks of filamentous proteins that are responsible for the spatial organization of cytosolic components. Inside microvilli, actin filaments form tight parallel bundles, which are stabilized by cross-linking proteins. While deeper in the cytosol, the actin network adopts a gel-like structure, stabilized by a variety of actin-binding proteins. Filaments, capped at their minus ends by a protein complex, grow away from the plasma membrane by the addition of actin monomers to their plus end. The actin network is a very dynamic structure, with continuous directional polymerization and disassembly. Severing proteins induce kinks in the filament and lead to the formation of short fragments that rapidly depolymerize or give rise to new filaments. The cytoskeleton includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules, causing their rapid plus-end depolymerization. Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane. The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles and microtubules. Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is partly dictated by their interaction with microtubules. All the microtubules originate from the centrosome, a discrete fibrous structure containing two orthogonal centrioles and located near the cell nucleus. Pores in the nuclear envelope allow the import of particles containing mRNA and proteins into the cytosol. Here, free ribosomes translate the mRNA molecules into proteins. Some of these proteins will reside in the cytosol. Others will associate with specialized cytosolic proteins and be imported into mitochondria or other organelles. The synthesis of cell-secreted and integral membrane proteins is initiated by free ribosomes, which then dock to protein translocators at the surface of the endoplasmic reticulum. Nascent proteins pass through an aqueous pore in the translocator. Cell-secreted proteins accumulate in the lumen of the endoplasmic reticulum, while integral membrane proteins become embedded in the endoplasmic reticulum membrane. Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane.
proteins to different parts of the cell. There can be no natural selection because unless all the parts are there, it will not work, and the cell will die.
Vesicles and microtubules.
To ascribe this structure to the random movement of molecules requires an incomprehensible irrationality.
It's been called one of the wonders of the molecular world. an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel